Hi, my name is Alan, and welcome to the first episode of HUSO Online Genetics. Today will be an introduction video, and we'll go into Mendelian and non-Mendelian inheritance by the end of the video. Today we'll also go into history as well, which would include Mendel's P's, so be ready for that. So firstly, we also want to see what we're studying, and Genex is the study of genes, and it's a part of your DNA that stores information that tells your body what kind of trait are you going to have. Are you going to have brown eyes? Are you going to have blue eyes? And so on. And it also studies heredity, which is how these traits are passed on through generations. And in this study, we also consider Gregor Mendel as the father of modern genetics, because in the 1850s he had a really interesting experiment with pea plants that was historically a huge breakthrough. Before Mendel, we had two theories, both of which Charles Darwin, that one, supported. The first one was blending inheritance. What would happen under blending inheritance is that the offspring of those two parents would have the exact average of the traits that the parents had. Now, this might make sense because you look at a population and you think, yeah, the kids look more or less similar to, I guess, a meeting point between the parents. But if you think about it on a more long-term scale, then eventually, if you just kept on breeding the same population over and over again, everyone would look the same because everything would average out over time. So naturally, that can't be it. And another theory that we had was Penginesis. Now, this is a little bit hard to understand, but you can almost think of it like a fax machine. What happens is in the gonads, they used to think that they would take a snapshot. So a snapshot of every single part of the cell in your body, we'll call those gemmules, and then those contribute to the gametes. And then after that, they fuse together. Now, this also is a really old theory that even Hippocrates was one of the first people to also propose this idea, but shortly you'll see why this doesn't work. So with Mendel in the 1850s, he's a monk in an abbey, and he decided, or a friar technically, he decided he wanted to take a look and see how genetics worked. And he noticed that pea plants, they had lots of cool traits like height, flower color, seed color, and seed shape. There's a lot more that he studied. He was actually also really lucky that he was able to avoid linkages, but we'll also go over that in a future class. So what he did was he took true breeding pea plants, and he did many experiments, but we'll talk about just one to do with flower color. They could either be white or purple. So he started with true breeding pea plants. Now, what that means is that these plants, if you just breed them with themselves down the line, consistently you always get just white flowers, or consistently you just get purple flowers. So what Mendel did was he painstakingly, and this is like no small task, this was thousands of plants, he hand pollinated the two lineages so they didn't cross pollinate across each other. Then he hand pollinated to intentionally cross pollinate the two lineages, so white with purple, and only that for every single offspring. And what we call these generations, just to make it easier to name, we call the parental generation P, so that's the first true breeding plants. And then the offspring that he created, we call those filial 1 or F1 generation for short. So now what happened? Basically, the first filial generation was only the dominant flowers, which was the purple flowers. And this seemed a little bit odd because at that time you have blending inheritance. You would expect, okay, purple and white will be pinkish, but no. They were all purple in F1. So that didn't stop him. He still hand pollinated, or in this case, he just normally pollinated all the F1 generation plants with each other. When he did that, he got the F2 generation, and this is where the Genex comes into play. So it turns out there is a 3-to-1 ratio. So 3/4 of the pea plants had purple flowers, and 1/4 of the pea plants had white flowers. And this doesn't really, I guess, make sense under the previous theories. And obviously, when you have new experimental evidence, that means that you always have to make a new theory, or at least adjust your old theories to incorporate the experimental evidence. So this is how we have genetics, where genes come in different versions. We'll call those alleles. So for example, you can have a allele for a white flower, an allele for a purple flower. So in this case, when the parental pea plants produce gametes, so like on the stamen and on the pistil, they will give one version of the allele that they already have. And we'll get into a lot of segregation in the future lesson. So in this case, every plant has two alleles. So it could be white-white, it could be white-purple, it could be purple-purple. In reality, I think it's easier if we think about it that way, but we'll call it something different in the next slide. So in this case, one allele, in this case, is purple, and that's dominant. And then the other allele, which is white, that's considered recessive. The thing is, if you have two purple alleles, then it'll always have purple flowers no matter what. Likewise, if your plant has white alleles, two of them, then it'll always have white flowers no matter what. But if you mix them, so you have one purple allele and one white allele, then the offspring the purple allele will mask the white allele, and then you'll have a purple flower. So the set of genes, so like in this case, we can call uppercase P, we'll call that the purple gene, and lowercase p, we'll call it the white gene. That's the genotype. So what are the alleles that they have? And then the appearance, so whether the flowers are purple or whether the flowers are white, that's what we call the phenotype. This is where our Punnett square comes in handy to understand the why we have a 3-to-1 ratio. And I guess I'll hand this over to future Alan to go over this problem with you. So this is an example problem where we're going to see both why there's a 3-to-1 ratio, and also how do we tell what the genotype is if we only know the phenotype that a plant is purple.
Alright, so right now we're going to draw a Punnett square, which is in this case a box with 2 rows and 2 columns, and then on the edges of the box we're going to draw the genotypes of each of the parents. So in this case they're both heterozygotes. Once we've done that, we're going to copy. So uppercase P, uppercase P, uppercase P, lowercase p, uppercase lowercase p, and lowercase p as intersection between the rows and the columns, which then gives us a ratio of 3 purple flowers and 1 white flower. But then the genotypes are going to be, 1 is homozygous, dominant, so the uppercase ones, 2 is heterozygote, and then 1 is homozygous for the for white, which is homozygous recessive, which is lowercase p, lowercase p. But what if we have a purple flower, we don't know whether or not it corresponds to the homozygous or heterozygous genotype? In that case, we can do a test cross. So we're going to take the situation where it's homozygous dominant, and we're going to cross it with a homozygous recessive. As a result, all of our offspring will be purple. So it's going to be a homozygous dominant purple flower. But if it were to be a heterozygote that was purple, that had a purple phenotype, then what would happen is you would see a 50% to 50% ratio of purple to white flowers, and then from that you'd be able to deduce that our initial purple flower was heterozygote.
Alright, so now we'll go on to non-Mendelian genetics. This is really fun, but it's also lots of different exceptions to the rule, which always it's got to be like that. So I'll just go over briefly each of them so you have an idea of what to look for. The first one is incomplete penetrance. So that's when, even though you have a dominant allele, other influences, such as environmental, can actually make the gene product, which is a protein, they can make those not necessarily the exact same as with different environmental influences. So that can result in things like polydactyly, and certain parts of the population are actually more at risk for that, depending on just genetic history. After that, we have incomplete dominance. So this one is one of the more common ones. You'll see that a lot, but basically you have a homozygote, which is 2 copies of the same allele, and you have heterozygote, which is different. So in this case, we have a dragon snap. You have a white allele, lowercase r, a red allele, uppercase R. And what happens is the heterozygote, so the mix of the two, you have one red allele, one white allele, one big R, one small r. So big R, small r. With that genotype, you don't actually get the dominant allele. It doesn't mask. Instead, it actually combines, so they're incompletely dominant. And instead, you have a blend of the two colors, which in this case gives you a pink dragon snap. Likewise, we've talked about two alleles so far, but you can actually have more, and blood type is a really common trait in humans that will typically have that has 3 or more forms. So in this case, you have antibodies and you have antigens. Antibodies will react to antigens, so you don't want to have anti-B antibodies in blood with B antigen. That's how blood transfusions in history tend to not always go down really well until we figured out what these were. These antigens are expressed on your red blood cells, and for certain people, they can have what we call an IA, an IB, and an I just lowercase I. So if you're blood type O, then that's 2 lowercase i's, you have no antigens. If you have IA and lowercase I, or 2 IAs, that'll give you type A blood. If you have IB and lowercase I, because IB and I are dominant to lowercase I, or you have 2 IBs, then that will give you type B blood. And finally, if you have both IA and IB, then you're going to have type AB blood. So that's how we get our different blood types. There's more rhesus monkey, but that's for another day. So there's also some problems that we have nowadays that are a result of this overdominance. So in this case, heterozygote advantage is where the heterozygote, so if you have one of each type of allele, is better off at surviving and reproducing than either homozygote. And sickle cell allele is a very interesting kind of genetic condition, because if you are homozygous for sickle cell, then your cells will have a hard time fitting through the small capillaries, and that can be an issue. But if you live in a place where malaria is endemic, then if you have the sickle cell allele, but just one of them, not 2, if you have just one, then you're actually going to have a better chance of surviving, because the malaria parasite will go into your red blood cell, but your cell will also basically lyse once that happens, and it'll give you essentially immunity to sickle cell. But if you are homozygous for not having the allele, then you don't have that. So that's why the heterozygote is actually advantaged in reproduction and survival. Next is, we haven't talked about chromosomes yet, but basically chromosomes are bundles of DNA where your genes are located on, and those genes encode for traits. So most of those are on autosomes, which are like 1 to 26, but there's also the X and Y chromosomes, and those are sex-linked. So if we have X-linked genes, and if you have a dominant copy and a recessive copy, and you're female, then it means that your dominant is going to mask the recessive. So that's going to be totally fine. But if you're male, well, females XX, you have 2 X chromosomes, but male, you see this sort of large X chromosome, but now you have a Y chromosome, which doesn't store too many genes. There are some that are on Y, but mostly it's on X. So if you only have 1 copy of X, now whatever copy of X you have, that's the only one you have. So that's why Duchenne muscular dystrophy is more likely to affect males than females, because males have only 1 copy to deal with. And then finally, we also can see how genes can affect each other, because genes are all located in the same chromosome. So through some feedback loop or other effect, then in this case, we see the gene for baldness will affect the gene for hair color. Likewise, the gene for baldness will also affect this gene for hair color. And that goes through gene expression, which we won't cover, so we'll just leave it at that. And then so this is kind of sad, but basically some alleles will cause mutations, and that will kill you. So a Agouti code is great, and so are yellow codes, but if you're homozygous for AY, well, you're dead. Sorry. And then finally, the last one is really cool, is snail shells, and that's an example of the maternal effect, where the genotype of the mother will actually determine the phenotype of offspring given certain genotypical compositions. So the picture is probably the best way to talk about that. Dextral, right-handed or right-shelled, and sinistral, sorry, left-handed people, but basically right-shelled and left-shelled. So what happens is we have homozygous and we have homozygous, and we cross-fertilize these two, so now they're the same. But if we take a look, so the mother provides the egg, so the maternal effect we see here, if the uppercase D, mother is the egg, then even if these two snail shells, if these two snails have the same genotype, their phenotype is actually different depending on what the mother had. So in this case, the mother is uppercase D, so therefore uppercase D is the one that is expressed, and we have a dextral shell. And then in this case, the mother has the lowercase d, so that expresses a sinistral shell. And then once they reproduce with each other, it just depends on the mother in the future. And it's the same 3-to-1 ratio that we've seen before. So as a result, we can see that traits are always inherited through genes, which in turn have different versions called alleles. And we can see that in Mendelian genetics, some alleles are dominant and some are recessive, and that's what affects phenotype. And then we also can use Punnett squares as a way to both understand the mathematical implications of probability in genetics, but we can't necessarily use that for everything because, like in real life, we don't have always Mendelian inheritance. There's always some traits that will behave in lots of complicated but also incredibly interesting ways. So I hope you learned something, and I'll see you in the next video.